Yeah, as David said, I'm a PhD student at Cardiff University and the Natural History Museum, London. This talk is called Snowball Earth, Ice Creatures of the Deep Past. I'm going to start by explaining what I mean by the deep past. So this is before human beings evolved, before the cavemen, before even the dinosaurs, before the first animals as we know it. There was still life going on in the earth. It can be hard to visualize this because it was such a long time ago. So how we often do this is by imagining it as a clock. So if you look at the clock I've got on the screen here, then imagine that at 12 o'clock, the earth is formed. Then just after 1 o'clock, you have the first ever life, which is kind of like bacteria, very simple and single cells. Then at almost 3 o'clock, it starts to use light to create energy. By 5 o'clock, we're getting more complex cell structures, kind of like the cells that are in us and in plants, animals, but they're still living as just tiny microscopic single cells. Then there's a long period of time which is known as the boring billion. And this isn't because nothing happens, but what most people are interested in is what comes next, the Cambrian explosion. This is where suddenly we start to find loads of animal fossils from this time, because after the Cambrian explosion, all of the kinds of animals that we know today come into existence. So shortly after that, you have the first land plants, you have animals coming onto land, you have dinosaurs, and then at just before midnight on our clock, you get humans. So that's quite strange that we have a really long period of life where things are just ticking along, being run by single-celled organisms, and then suddenly all of these animals appear. So what happened in, let's say, the hour leading up to this on our evolutionary clock? If we put this on a geological timescale, this is what geologists use to measure time. It's between the Tonian, where everything was still single-celled and very simple, and the Ediacaran, where we start getting the first animal fossils, and then shortly after the Ediacaran is the Cambrian explosion. So MYA on this little chart stands for millions of years ago. So it's between 720 and 635 million years ago. This is a time period called the Cryogenian. The reason that it's called the Cryogenian is that cryo is another word for cold. This time period is also known as the Snowball Earth because the whole planet was entirely frozen all the way to the equator. This is different from the ice ages, like the ice age where the mammoths lived, because that's just the poles coming a little bit closer, a little bit further down. This is one of the very few periods in history when the entire planet is frozen over. And life had to not only live through this, but it had to be diverse and supported enough so that not long after, all of the animals as we know it today evolved. So my research question is, how did life survive on a Snowball Earth? I'm going to break this down into the topics I've been looking at. What key habitats supported life? What were the kind of oasis of life in this hostile world? What kind of organisms lived there? And how can we replicate these habitats in the lab so that we can actually prove that things were able to live there and survive? And then can we imagine a whole Snowball Earth ecosystem? Because small isolated habitats can't really explain how a whole planet worked. So those are the 4 questions that I'll be going through in this talk today. So let's start with the first one. What key habitats supported life on Snowball Earth? What we can do to start to answer this question is look at how life survives in the ice today in the polar regions, the Arctic and Antarctica. Here are some examples of places where we found life in the poles in the Arctic and in Antarctica. So like anywhere else, these places are surrounded by sea, particularly the Arctic is an ocean area. And so things live under the sea. They're particularly concentrated in places like hydrothermal vents, which you might have seen on David Attenborough documentaries. They're very warm, so life tends to cluster around there. However, this can't support all of the diversity of life of Snowball Earth because it requires a lot of photosynthesis things like algae, plankton from the surface to be able to feed everything at the bottom. And if the whole ocean was frozen over and running out of oxygen, then that system would fall apart. Similarly, there are lots of streams under the glaciers in the Arctic and the Antarctic that are full of life, but not the diversity of life that you would need to support a whole ecosystem, because again, not much sunlight means not much photosynthesis, which means not much of the plant-like matter that things need to live on to survive. So a lot of the algae is under the ice in algal blooms and on top of the snow. This is a great source of food filtered into the ecosystem, but again, it in itself cannot support a whole community. These are usually just dominated by one or two types of algae rather than being a whole ecosystem. The last habitat is something called a cryoconate hole. I'm going to focus on this partly because you might not have heard of this before, so I'm going to explain it a bit more, and partly because this is a habitat that's particularly interesting to Snowball Earth researchers. Cryoconate holes are small, little, tiny meltwater puddles. When you get dark, organic and inorganic matter that accumulates together on the snow, because it's a darker color than the snow, it absorbs more radiation from the sun and melts down into the water, creating a little pocket of meltwater. These are sometimes just a few centimeters wide, like this in this photo, but they can be much bigger. Cryoconate holes are particularly interesting to a Snowball Earth researcher because they're on the surface, so most of them can support photosynthesis. They could still survive if the ocean became inhospitable, if it was frozen over, if it was running out of oxygen. And researchers have found that even when these cryoconate holes completely freeze up in really cold, hostile conditions, things still survive in there. So that brings me on to the next question of what organisms lived in Snowball Earth habitats. So we can look at what kind of organisms live in cryoconate today, and that's what I did a couple of years ago. I collected up cryoconate from both polar regions, from the Arctic, Sweden, Svalbard and Greenland, and from the Antarctic. The bigger map is just a zoomed in view of the McMurdo Dry Valley site, which is on one side of Antarctica, near to the coast. And then I also have some samples from the Estinen Scoop, which is the other side of Antarctica in Queen Maud Land, and is much closer to the mountains. Most of these samples were collected by my very kind supervisors and some collaborators in Leeds, but I did go and get some myself from Svalbard, an island in the Arctic. That's my foot there for reference of what a cryoconate hole in the Arctic looks like. But you do have to be careful when you're sampling with your head down in the Arctic, because there are polar bears just round the corner. This photo was taken just the day before by my friend Roslyn when we were out on a boat. So it was an exciting trip. So once we've got these cryoconate, how do we figure out what's living in there? We go through a process called DNA sequencing. And I carried this out in the Life Department at the Natural History Museum with my supervisor, Anne Youngblood, because they have an excellent in-house DNA sequencing facility. How this works is we collect the cryoconate, and through a chemical reaction, we extract the DNA, which is the code of living things. We then get a particular piece of DNA, a gene, which is slightly different in every species, so it can be used to tell them apart. We copy this lots and lots of times so that the machine can detect it. We then clean it up so only that gene remains, again, so that the machine can just read this one gene. And I did this twice. Once for a gene that will identify simple-celled organisms like bacteria and archaea, another type of single-celled organism, and then a second gene which will identify the more complex organisms like algae and fungi, things like that, and amoeba. So then what we're left with is a code, and it's slightly different for each species, so we can refer to a database to see what kind of organisms are in the cryoconate holes. What I found was a huge diversity of life. I found 35 phyla, which is the most general group of organisms, which means, tens and hundreds of species. There were the algae, such as chlorophyta and rhodophyta, There were choanoflagellates, which are like a really early animal ancestor. There were bacteria, types that live in the air, types that like no oxygen, types that use light. There were protists like amoeba. There were even some tiny animals like the tardigrades, which have got a bit of a reputation for living in all the extreme environments on Earth, tiny little animals. So we now know that Cryoconate over both of the poles is an oasis for incredible diversity of life compared to other cold climate habitats. Not only that, we can go back to our evolutionary timeline. We know what kind of organisms had evolved by that point, and we can compare it to modern Cryoconate. So we can not only see that Cryoconate has a load of different organisms in it, we can also tell that they're very similar to the organisms that would have been alive and important back then. Things like protists, algae, choanoflagellates, all needed to survive through Snowball Earth. So it's really exciting that we find them in Cryoconate. That's good evidence that Cryoconate could have been an important ecosystem for preserving life through Snowball Earth. Not all Cryoconate is made equal. The Arctic holes and the Antarctic holes are quite different because the Arctic and the Antarctic are quite different places. The Antarctic is on a landmass and is much drier. The Arctic is made up of islands and sea ice, and it has different impacts on the environment. So the Arctic Cryoconate holes tend to be like the ones on this picture on the right. They tend to be open at the top. They tend to have these sticky granules at the bottom of them. The Antarctic Cryoconate holes are covered by an ice lid most of the time, and they have a thicker, sandier layer at the bottom, the samples I've been looking at. And this means that they have different organisms in them. There's a divide between life of the Antarctic and the Antarctic, which isn't that surprising given they're very far away from each other, and they are quite different places. So not only do we know that Cryoconate in general is similar to life on Snowball Earth, we should be able to tell which kinds of Cryoconate are most similar to Snowball Earth, which have most of the same organisms. The conclusion I landed on was the McMurdo Dry Valleys in Antarctica. They were what resembled Snowball Earth life most, and you can kind of tell why by looking at this picture. It looks like a whole snowball planet. So now my third question. The thing is that the McMurdo Dry Valleys are not the same as Snowball Earth. They have similarities, but the Earth would have been so cold back 650 million years ago that most life would probably have lived on the equator, where there was the most melted water. Where the poles are now would have been far colder than the modern polar regions. So that would mean that rather than the full-time light in the summer and full-time dark in the winter that Antarctica has, you'd have a 12-hour day-night cycle like you have at the equator. So what we do as scientists is if we can't find a system that's exactly the same, then we try and replicate it in the lab. Fortunately, at Cardiff University's Earth and Environment department, I have these two really cool incubators where I can control a lot of the conditions. I can control the light, I can control the temperature. So what I did is I got the same pool of Cryoconate, I put it in these little bottles which are about the same size and shape as the Cryoconate where it was found. And I had one incubator running at normal Antarctic conditions, and then another one which I put in Snowball Earth type conditions. So like I said, at the equator, you've got 12-hour day, 12-hour night. So the first one I did was 12 hours of light, 12 hours of dark over a period of 28 days. And I compared it to just Antarctic summer, which would be light pretty much all the time. And so you can see this top graph is the results I found. And the Snowball Earth conditions and the modern conditions are very, very similar to each other in the results. The results show air saturation, which is basically a rough measure of how much they're breathing, the organisms are respiring. So it's a good indicator of how much the community is growing. And I have a control system which is no light. So you can look at Snowball Earth conditions compared to modern conditions. They're very similar, and no light is a different case. So to me, that says that even if you took a cryoconate hole from the modern Antarctic and you put it on the equator in Snowball Earth, it would behave in a similar way, which means that the Antarctic is a good model system for Snowball Earth. It's a good sort of analog. Then in the second graph below it, that shows when I did the same thing with a freeze-thaw cycle. So I did 12 hours of thawing and 12 hours of freezing compared to modern Antarctic conditions where it's pretty much the same temperature. And I ran this for, I think this one was 37 days, 35 days or 37 days. And again, the Snowball Earth conditions and the modern conditions ended up at around the same place, and they followed about the same pattern. So that means that, again, if you take a modern Antarctic cryoconate and you put it in this kind of equatorial Snowball Earth type condition, it behaves in the same way. So it's probably a good model system. If we study cryoconate now, we can see some insights into what Snowball Earth life might have been like. So that was that third question. Now my last question. These little tiny pockets are really interesting and important, but in the real world, life doesn't work like that. All of the habitats in the world are connected somehow. The cryoconate has got to come from somewhere, and when it melts, it has to go somewhere. So how can we imagine what the whole Snowball Earth looked like? How I did this is I traveled just before lockdown back in February, all the way down to Antarctica. I went to Signy Island off the coast of Antarctica. So we flew down to Rothera, which is the UK's big research station, and then we took the ship over to Signy Island. We stayed in the base, which you can see a picture of there, which is down by the coast. Also down by the coast are an awful lot of seals and penguins. It's a very busy place. But if you just go a little way inland, then you see the closest that I have ever seen to what Snowball Earth would be like at the equator. There are flowing, changing, interconnected habitats. There are streams, lakes, there are blooms of bright pink and bright green snow algae. And what I did was I took samples from all of these distinct ecosystems so that when I get them back to the lab, I can look at the similarities and differences. When the snow melts out, do those things survive? If a cryoconate hole, flows into a stream, where do those organisms go? Which of them survive? So hopefully by looking at this little island Snowball Earth system as a whole, then I can get a better picture of what Snowball Earth as a planet would have looked like, as a interconnecting habitats that are all related to each other and have preferential conditions for some organisms and probably not others. So that's how I have been addressing the question of what a whole Snowball Earth will look like together. And I think it would probably look something like this. This is a picture I took on Signy Island in March. Thank you very much to the British Antarctic Survey, who took me on this trip as part of their collaborative Antarctic science scheme. And thank you to you all for listening. That was, it was really fun to talk about my adventures. Thank you to the NERC, the Natural Environmental Research Council, who fund my research, and for Cardiff University and the Natural History Museum who host me. Thank you to my supervisors, and thank you again to David for organising this and introducing me.
Well, not at all. Thank you very much, Jas, for a very interesting talk and quite spectacular and uncommon sceneries. Was it really cold there when you went there?
I am quite a cold weather person. When I first got there, it was the middle of Antarctic summer in February. And so quite honestly, they gave me all this great gear. They gave me these big coats and waterproofs, and I just stripped off to the level of wearing a shirt and leggings. But by the time it was sort of March, April time, then it was getting snowy and colder.
Yeah, I can imagine. Probably very, very cold. So as usual, I'd like to invite the people here to ask some questions. If you've got some questions, so you can use the Q&A option, So you can just write down your question, and I can read it out loud, and then we can have a discussion going on. So I don't know if there is any question. I cannot see anything right now. I have a question, but I don't want necessarily to be the one starting the questions. Okay, right. There are a few questions now. Great.
Mm-hmm.
So first question is, how do the I guess photosynthetic bacteria survive in the Arctic and Antarctic winter when it's 24 hours of darkness and they can photosynthesize? Do they have stores of energy from the summer, or do they have a dormant state like a hibernation?
That's a really good question, thank you. Yeah, it's pretty much like hibernation, dormant state. So this is particularly why I described algae as coming in blooms, and cyanobacteria, the photosynthetic bacteria, sort of do the same thing as well. Over the summer, you get loads of them growing, and then over the winter, they're still around. Some of them might be killed off, some of them will be dormant, but there will be enough there by the end of the summer that as things start to darken, the heterotrophs, which means the things that eat other things, will sort of take over. And then by winter, the metabolic rate, the rate at which the cells work, slows down, which is what happens with lower temperature. Whether they're entirely inactive when they're at their coldest, I think, is a question for some different cryoconate hull researchers, but it's definitely a question in the research community. But we do have to recognize that when we collect cryoconate and study it, we are just looking at a certain snapshot. What I can say is that my cryoconate samples, some of which have been in the freezer for 10 years in the dark, when I start growing them again, the cyanobacteria and the algae pops up. So clearly quite a lot of stuff can survive a really long time in the dark and in the cold.
Thank you for the answer. I think, yeah, you've well addressed the question. There is another question here. First of all, that's a comment. Interesting talk, thank you. And the question reads, I wonder though if anyone else had problems starting up. Oh, right, that's actually more like a technical question. I should have read this. So apparently some people have had some problems with the login this time. So I'm really sorry about this. That's the first time that it seems we have a problem about this. So just to clarify the situation on our side, normally we start the actual event 10 to 15 minutes before the actual start of the talk. So I'm really sorry if you could not join us. I'm afraid that's probably related to the Zoom system, so we don't have a full control on this. So I can only encourage you to perhaps try to log in a few minutes before. Maybe it's because a lot of people are logging at the same time. I don't know. We never had that problem before, and we've run in the school, for instance, sometimes meetings with nearly 100, 120 people. So I don't think it's related to the number of people. So I can only apologize for this, and I hope next week is going to be better. If you still have some problems, so please let us know, and I'm going to talk to our IT to make sure that it doesn't happen again in the future. So let's go back to the questions. So yes, so there is a question about the preservation of the cryoconites. How do you preserve the samples before the analysis?
So some of the samples were preserved chemically with RNA later, which is a chemical designed to preserve RNA and DNA, the genetic components, but most of them, we just kept them really cold. Most of them are frozen, and the best thing to do is to freeze them as quickly as possible and as cold as possible, and then keep them at sort of the same temperature. So that's what we do most of the time. That seems to be adequate. We don't see big difference between chemically preserving things and not. And sometimes it's better to keep them sort of pure without chemical additives.
Yeah, great. Another question, it's a question from Rowan. How did the Snowball Earth happen? How did it start?
That's a really good question, Rowan. And I'm really sad I didn't have more time to talk about that really, so I'm glad you asked. How the Snowball Earth happened was a matter of climate change. So the gases in the atmosphere were changing, which allowed the poles to sort of creep down and get bigger and bigger because of the concentration of greenhouse gases and how the kind of volcanic under the earth things were happening at the time. The poles got bigger. Because the poles are white with the ice, then they reflect sunlight away, meaning that it gets colder and colder. The more ice you have, the more ice there becomes, because white things stay cooler than dark things, like with the cryoconate. So this created what we call a runaway albedo effect. Albedo is the reflection, so it kept just creeping further and further and further down until it was all the way down to the equator, where it stayed frozen for a long time, until this was disrupted, partly again because of greenhouse gases in the atmosphere, which were changed by the organisms living there, and also possibly some volcanic activity melting and creating ash.
Great, thank you very much. There is another question. That's it's a bit looser as a question, but perhaps you can comment. There's someone thinking that there might have been several iterations of Snowball Earth, and the question is, were they all similar? Or...
So yeah, there may well have been more occasions when the Earth froze completely to the equator. I believe that most of these all of these would have been before what we call the rise of algae, which is kind of the lead up to animal life as we know it. I think it's entirely possible that some of the first animals, which would have been primitive sponges, may have evolved during Snowball Earth or immediately afterwards. And there was certainly lots of algae forming sort of structures together. So I wouldn't say that previous Snowball Earths were biologically similar. Bacteria may have employed similar ways to survive it if those previous Snowball Earths happened, but it wouldn't have needed to support such a complexity of life as the Neoproterozoic Snowball Earth would have done.
Great, thanks. Another question. We've got a lot of questions. I don't know if we can go through all the questions. I've got the whole evening, but yeah, well, anyway, that's the advantage of a webinar. People can very gently disappear out of the virtual room. Anyway, so I'm going to read the next question. What were the granules in the holes?
These are basically cryoconate that is formed together in balls by sticky extracellular proteins mostly. A lot of the cyanobacteria produce these. Some cells sort of exude chemicals from them, which can form biofilm type things that hold organic matter together. So that's what the granules in cryoconate are, to the best of my knowledge.
Okay. A question from Trevor. Are these conditions, presumably cold conditions, similar to what might be found on icy moons?
Yeah, that's a really interesting question. I think that it might be. If the moons have winds, then that's important. I think it's important to have an atmosphere, and not all moons have as much of an atmosphere as the Earth does. I know that some people who are interested in Mars and Martian biology are quite interested in my research and being able to sort of simulate icy life on ice surfaces. So I wouldn't be surprised if people who look into icy moons are starting to think the same things too, as long as they have enough of an atmosphere to move all the organic matter around.
Yeah, great. Another question is about the nature of the organisms. Are there any organisms that use a chemosynthetic way of producing energy, or a bit similar to what we could have around hydrothermal vents?
Yes, there are quite a lot of mixotrophs which use chemical methods and photosynthetic methods. The algae, I think the Chlamydomonas and some of the other algae that are really dominant use both methods. I'm sure there are some purely chemotrophic ones as well. I know there are some cryoconate researchers who look at chemolithotrophs.
Okay, cool. Another very positive comment, so really great talk, Jas. Does this research lead us to any deeper understanding of modern life that lives in extreme environments?
Yes, so one of the things that I found and am writing about at the moment is actually not really directly linked to Snowball Earth, it's just about the diversity of life on the glacier surfaces, because the techniques I was using for DNA sequencing are relatively new. So the study I did is definitely the first, or one of the first, big Arctic and Antarctic DNA surveys of the ecology. So we're able to see in a lot of detail just how many organisms are living there, which I think is quite important.
Okay. Another question, totally different. How did the extinction rate vary through the Snowball Earth period? You didn't really talk about extinction, but...
Yeah, so that's quite difficult to say, because we don't really have so much of a fossil record as sort of DNA molecular clocks, where you trace back, you have two organisms that have a certain number of differences in their DNA. And so you trace back how long that would have taken as to how long ago they sort of separated from each other. So unfortunately, that means you can only base things off modern living organisms, and you don't have much record of stuff that went extinct. However, what we have found is that there's no particular evidence of what we call a bottleneck, where suddenly there's a sort of mass extinction which leads to diversity. It looks like the diversity that comes towards the end of Snowball Earth is not the kind of thing where there's a big mass extinction, so therefore there's loads of like opportunities for organisms to fill niches and habitats. It doesn't look like that happened. So that means that just a lot of stuff must have been able to survive Snowball Earth somehow.
Great. How accurate is the gene multiplier that you've used for the gene sequencing? Because, well, some people, we can imagine that sometimes you get small mutations. So is there a way to screen these out?
Yeah, we do go through a filtering and screening process. So sometimes bits of DNA can stick together, or sometimes sequences that you don't want or haven't been cut in the way they're supposed to be can still remain. And so when you get the results from the DNA sequencing, then you can go through a bioinformatic process, a process using computer software, where you can filter out things that look really, really different from what you're expecting, because you know how long the piece of DNA you're expecting is. So if there's something that's dramatically different, then you can filter it out in that step.
Okay, great. Yes, were there interglacial periods during the cryogenian, so that Snowball Earth period?
Yes, there was one really significant interglacial period. So it looks like Snowball Earth happened in two very large glaciations. So there was the Sturtian and the Marinoan, and there was probably a period of melt between these two. So you have, like a 30 million year glaciation, and then a big melt, and then a 60 million year glaciation. I don't know the chronology very well, unfortunately. I haven't memorized it, but basically there's a smaller glaciation and a bigger glaciation with a melt in the middle. I imagine there was probably still melt times in much smaller scales than that, because that's how the climate tends to work. It's one of those things where modelers of the Snowball Earth are working on this and trying to get it as exact as they possibly can. But it looks like mostly two huge glaciations.
Yeah, okay, great. That's an interesting one, kind of timely one. Are we losing cryoconate holes due to climate change?
Yes, I imagine we are, and we're probably gaining some as well, because they tend to appear quite a lot in the melt areas, the ablation zones, the melt areas of glaciers. So there is this thing with dirty ice, because like I said, when ice is a darker color, it melts faster. So organisms are creeping onto the glaciers, and they're getting covered in things like cryoconate, they're getting covered in mosses and lichens. The tundra is creeping up to the Arctic, and the forest is creeping up to the tundra. And when that happens, the ice will only melt faster because it's getting darker. So some glaciers are starting to melt completely and we're losing them. So we're losing cryoconate holes. Some of them are becoming dirtier and we're gaining this, but that is not a good thing even as much as I love finding cryoconate, because it means that the poles will accelerate. It's the opposite of how Snowball Earth was formed, where I got whiter and whiter and whiter. The poles are getting darker and darker and darker. So microbiology is really important for understanding climate change. And that's what my supervisors group in Aberystwyth look at quite a lot.
I see. There is a question about the preservation of ancient species here. I think we can kind of translate a little bit that question to, I mean, is there any evidence for fossils, microorganisms that could have lived during the Snowball Earth period in environments similar to those that you're studying today?
The difficult thing about glacier surface habitats is that they melt. And so some people struggle to be convinced by life on ice when they're used to hard evidence in the terms of fossils. We do have fossils from the time period. These are usually fossils of microbial mats. So you have a sort of lots of tiny little blobs all stuck together. The problem with fossils from this period is that we can tell that it's something that was alive, and sometimes we can even tell if it was bacteria-like or eukaryote-like, but we can't tell any more than that. So that's where the DNA molecular clocks looking at differences between different modern organisms comes in.
Yeah. Very good. I think we can take the last couple of questions. Yeah, so are there more cryoconites forming from carbon particles that might actually be produced during the burning of fossil fuels? What could be actually the source of the materials going to make these dark patches to create the cryoconites? And I like this question. I had a very similar question actually to ask you, because what you're looking at today is obviously an open system, and you could get dust being carried all around the world, and this dust at some point is going to accumulate in small, you know, ponds or small kind of volumes of water on the ice. Yeah, so do you have any feeling about this? Do you think that what you're looking at today is mostly influenced by, you know, far traveled dust? Or do you think that this could also be related to more minor weathering processes in Antarctica, for instance?
Yeah, airborne transport is really important, and it's where a lot of the matter for cryoconate comes from. Yes, it is possible that man-made activity could influence the composition of things like cryoconate and sort of glacial habitats. There are limits on how far things can travel through the air, which is a sort of another area of study. I think that, yeah, it's interesting because cryoconate sourced from a lot of places. It sort of comes flowing in streams. A lot of it comes from the air. I think that if the planet continues to warm and humans do start settling further north and further south, we might see more impacts from it. I do know that the Arctic and the Antarctic bases try and be really careful with how much pollution they produce because of this. When I was on Cygni Island, I was working with someone who was looking for microplastics in Antarctica, because these things do travel. A lot of different types of pollution, a lot of different types of carbon, silicon do travel. There are limits to it, but yeah, that's a really interesting question. And I think that if you were doing some chemical analysis, you might be able to tell where different chemicals came from.
Great, thank you. And as a concluding question, sorry to ask you so many things. You had actually quite a bit of interest. It's great. So yeah, what future research are you interested in?
That's a difficult question. I'm interested in a lot of things all of the time. I'm interested
to answer if you don't have a clear answer.
I'm interested in, like I said, I've been talking to people who work on Martian systems. So if I could replicate the same experiments I'm doing for a Martian environment to see whether hypothetically microorganisms could tolerate a Martian environment, that would be really interesting. I'm interested between the borders between the glaciers and the tundra, things like it looks very barren. When you first go to the Arctic or Antarctica, it looks barren because you're used to seeing trees and grass. But actually, once you've been there a while, you start to look closely and you see tiny mushrooms this big, and you see flowers and lichen. It's just that they're all about this big. And I find that really interesting. And so maybe I'll branch out from microbiology into some slightly bigger organisms at some point, but really just microbes in extreme environments. So I've done really hot environments before for my master's. I've done really salty environments with the Marine Biological Association. So maybe I'll find a new extreme environment I haven't done yet, like things that live in rocks, lithophiles maybe.
Very good. Yes, definitely. There are still many weird places to visit, that's for sure.
Yeah.
Either physically or intellectually through research. Yeah, Mars is definitely a very promising avenue. And I'm sure there could be some connections between what you've done so far and what is currently being investigated on the red planet.
Yeah.
Well, thank you very much, Jas, for this very interesting talk, and thank you all for staying until the very end and for asking so many questions. The talk next week is going to be again in a slightly different place. We're going to go to Africa, to the East Rift, and we're going to talk with Jack Williams a little bit about how to make forecasting of earthquake using geological information. So thank you very much all for a very good event today, and hopefully see you again next week.
Thanks very much.
Goodbye.
Bye.